Hannah Donnert with the Collaborative on Health and the Environment. Che enjoys bringing the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everyone to today's Che EDC Strategies Partnership webinar, which is titled Data Integration, Analysis, and Interpretation of 8 Academic CLARITY-BPA Studies. Our moderator today is Cheryl, Director of the Commonwealth Biomonitoring Resource Center. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the top of the menu bar on your window at any point during the presentation. After the presentation, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period, and we'll follow up on unanswered questions after the webinar. For those of you who have called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone in the webinar right now is muted with the exception of our moderator and our speakers. The webinar is scheduled to last for 60 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Cheryl.
Thanks very much, Hannah. It's my great pleasure to introduce today's outstanding panel of scientists. They're all EDC pioneers who have carried out groundbreaking research concerning human health and early life exposures to EDCs. Today, they'll be describing their research under the auspices of the CLARITY-BPA program. Their research clearly indicates that current regulations are inadequate for the protection of human and ecosystem health. Our first presenter is going to be Dr. Jerry Heindel, who will give an overview of CLARITY, its goals, processes, and outcomes. Dr. Heindel is a former scientific program administrator for the Division of Extramural Research and Training at NIHS, amidst many other positions there, and currently heads up HEEDS, the Healthy Environment and Endocrine Disruption Strategies Group, which he founded and currently directs. Dr. Gail Prince is the Michael Reese Professor of Urology and Physiology at the University of Illinois at Chicago University and the co-director of the Chicago Center for Health and Environment in the UIC School of Public Health. Her basic research focuses on estrogen actions, including the influence of early life exposures to EDC chemicals and toxins on adult prostate cancer risk, and that will be a topic of her presentation today. Dr. Anna Soto is an internationally renowned leader on environmental health and a pioneer in the field of endocrine disruptors. Her research discovered that fetal exposure to bisphenol A, a chemical often used in plastic products, which she discovered, produced deleterious effects in females, such as obesity, altered behaviors, decreased fertility, and mammary cancer. And her topic today will be a discussion of mammary gland cancer and EDC chemicals. Dr. Cheryl Rosenfeld is a professor at the Biomedical Sciences at the University of Missouri and at the Institute of Data Science and Informatics. She edited and co-authored the book titled The Epigenome and Developmental Origins of Health and Disease, otherwise known as DOHAD, and is president of the DOHAD Society. Her laboratory continues to use pioneering omics and bioinformatics approaches to reveal the mechanisms by which exposures to EDCs lead to health disturbances. So we will begin with Dr. Heindel and his overview.
Thank you very much. My goal today is to provide an overview of the CLARITY-BPA program as an introduction to the manuscript we're presenting today, the manuscript being Data Integration, Analysis, and Interpretation of 8 academic studies in the CLARITY-BPA. So CLARITY stands for Consortium Linking Academic and Regulatory Insights into the Toxicity of BPA. This is a new collaborative model, and the goals of this model are to integrate the strength of a guideline study with the molecular and disease-associated endpoints from independent scientists into one study. The result of that should be to improve safety assessment. So the partners in this CLARITY-BPA program are the NIEHS, both the National Toxicology Program and the Extramural Grants Program, and the FDA and 14 NIEHS-funded academic grantees. So why did we set this up? The current approach to risk assessment really focuses on guideline studies. These are studies with a standard design, standard endpoints, and a government-approved protocol. So if there's one or two guideline studies, the regulatory agencies depend on those to make their decisions, and they usually don't pay hardly any attention to the independent sciences studies, of which there may be dozens, hundreds, or even thousands. These are studies with a diverse design, diverse endpoints, and fewer animals per group. So to solve this problem, we came up with the CLARITY, where we would merge the guideline studies and the independent studies into one study design. So there would be a core guideline study with additional animals added for the independent scientist studies to use. That would be a government-approved design and more comprehensive endpoints of toxic effects. So let's talk about the study details. This study used a Sprague-Dawley rat. The core study had 50 animals per dose, and all of the independent scientist studies had around 10 animals per dose. There was a vehicle control and 5 doses of BPA going all the way from 2.5 micrograms per kilogram body weight to 25,000, and a positive control of ethynyl estradiol. The animals were gavaged from gestational day 6 to birth, and then starting again on postnatal day 1 to day 21, or every day for 2 years. So what we have is a continuous study with dosing every day and a stop dose study where the study dosing ended at postnatal day 21. And the core design took endpoints at 1 and 2 years, but the independent scientist studies you're going to hear about today took endpoints at day 1, 15, 21, 90, 180, and 1 year. The core study guideline endpoints were clinical chemistry, astrocyclicity, sperm parameters, body weight, histopathology. But the independent scientist studies, of which there were 14, if you look here, you can see they looked at many different organs and many different diseases like obesity and diabetes, breast and prostate cancer. So where are we now? The study is over. The core guideline study has been published, and 10 of the 14 independent academic studies have been published. The final goal here was to have an integrated publication of all the data, both from the core study and the independent scientist study. But it looks like the FDA is not interested to actually do that. So we decided to do an integrated publication of the independent studies to pull all that data together. So this is a very unique manuscript that we're talking about today. It's a final report of 8 that presents their data from the CLARITY and puts it in perspective to the previous published data they produced on BPA. And in addition, it uses new statistical analysis to integrate all of the findings and interpret them as a whole. So there were 14 studies, 10 of which were published. 2 of the studies, the testis and immune function, although published, decided not to participate. And there were 4 studies that are not yet published. So what we had is 8 studies, which you can see here. genital, cardiovascular, gland, ovarian, thyroid, learning and behavior, behavioral changes in brain transcriptomics, and then we added data integration across organs, doses, and time, all into one manuscript. Now, for each of the organ systems, which there were 8, the manuscript is set up to look at what were the previous publications to show an effect on that organ, then the hypothesis tested in the CLARITY study, methodology, a summary of the data, and then a discussion of the major findings, and comparing those findings to the previous publications, and when they didn't agree, perhaps sometimes, to look at study differences. Then there's a section on data integration across the organs, doses, and time, a discussion of that, and a final integrated discussion. So we couldn't have time today to present all of the data. So as you heard, we're going to present two data analyses, one on the prostate and one on the mammary gland. And then we're going to present the integrated assessment across the organs, doses, and time, and then come back and have a summary and the key messages. So with that, I'll turn it over to the other speakers.
Okay, thank you very much, Dr. Heindel. And we're going to go now to Dr. Gail Prince, who's going to talk about the effects of EDCs on prostate. And just while she's getting her slides up, remind everybody to type your questions into the chat function, and we'll address those questions after everyone's presentation. So Dr. Prince, I leave it over to you.
Okay, thank you very much. So I'm going to talk about the prostate findings that were derived in our laboratory for the CLARITY. And by way of introduction, I want to have a slide giving a background to why we're looking at this. So my laboratory has been looking at the effect of early life exposure to estrogenic chemicals for several decades now, and how that influences prostate disease with aging. We previously showed and published extensively that exposure to estrogens themselves, if they're high enough, can directly drive prostate cancer in adulthood. So for those studies, we utilized the rat Sprague Dawley, the Sprague Dawley rat, and we decided to utilize the same models and test it for bisphenol A. And the reason we use the rat is because it's a particularly nifty model in that the prostate gland develops entirely during the first week of life, just after birth. And this is analogous to in utero development in the humans. And so what we did with our BPA studies, there were many of them. We looked at a variety of doses, but mostly focused on the low dose of 10 micrograms per kg body weight, because that is relevant to human exposures. And we gave those exposures on days 1, 3, and 5 of life. We either did oral exposure or we did subcutaneous depot injection in oil to allow slow release throughout the day. We didn't really see differences in those two exposure methods. And what we found when we aged those animals and looked at it was that BPA at these doses examined was not sufficient to drive prostate cancer with aging. So what we did next was we looked at an additional exposure to estradiol in adulthood in these animals, a second hit, if you would. And the reason we do this is in aging men, the serum estradiol levels increase steadily, while the free testosterone levels decrease steadily, shifting the E to T ratio in favor of estradiol. So what we did in our animals is we gave them implants for a constant physiologic level of testosterone and a 2-fold higher level of estradiol. And when we did this, what we found was that in animals just treated with vehicle in adulthood, there was this very low incidence of prostate cancer. But if they were treated neonatally with BPA, there was a significant increase in prostate cancer incidence, severity, and progression. So what we did in CLARITY-BPA is we tried to first replicate these findings using the model system at the FDA. It was somewhat different. We used the stop dose animals where the exposures started at embryonic day 6 and were finished at postnatal day 21. In adulthood, the animals were given a steady dose of physiologic testosterone plus a 2-fold higher level of estradiol, and the animals were euthanized at 1 year of age. You can read our publication to see the details, but in summary, we were able to largely replicate our previous findings. There was an increased prostate cancer risk if they'd been exposed developmentally to BPA. Here we're looking at the estrogen-sensitive dorsolateral prostate intraepithelial neoplasia. And we can see that if they were treated with estradiol or any of the doses of BPA neonatally, that there was an increase in the severity score, putting it in the range of high-grade neoplasia, the precursor lesion to prostate cancer in humans. And the greatest effect was at the lowest dose of BPA. Furthermore, when we looked at cancer itself, adenocarcinoma in the dorsolateral prostate ducts, what we found was a striking increase in the number of tumors that developed at the lowest dose of BPA. So in the control animals given T plus E in adulthood, there was an average of 1 tumor per animal that appeared. But if they received this very low dose of BPA, we saw anywhere from 5 to 8 tumors per animal, seen here at low power, medium, and high power, where we can see the epithelial cells invading. And so we see this somewhat at the higher doses as well, although the N numbers were too low to determine significance. So in the second portion of CLARITY, we asked the question, how might this early life BPA exposure affect prostate cancer susceptibility later in life, long after BPA is cleared from the body? So we turned our attention to looking at the stem and progenitor cells of the prostate. In this schematic, we're looking at prostate epithelial hierarchy. In both the human and the rat, the prostate is a simple columnar epithelium composed primarily of secretory luminal cells lying on top of basal cells and rare neuroendocrine cells. And these are derived developmentally and maintained throughout life by long-lived rare stem cell population that occasionally undergoes asymmetric division, replicating itself, and giving rise to a bipotent progenitor, which then transiently amplifies, and during these rapid divisions, lineage commits either to a basal or luminal progenitor. So we asked whether early life exposures or continuous exposures to BPA might be influencing some of these decision points. So the model was the animals were treated at the FDA. We only looked at the low doses, the 3 lowest doses of BPA and estradiol. They were gavaged daily from embryonic day 6 to the mothers, every day of life through 6 months of age. When the animals were sacrificed, the dorsolateral prostates were placed on ice, rapidly shipped to our laboratory where we dissociated the epithelial cells and put them into a matrigel culture. And in this culture system, most of the epithelial cells are not capable of surviving, but the prostate stem cells not only survive, but they start to rapidly proliferate. And by day 7, you see these small prostospheres consisting of stem and progenitor cells. So at 1 week, we collected and dissociated them, transferred them to a new culture. After another week, we transferred them to the 3rd culture and then examined the endpoints after 7 days. So this is 21 days without any exposure to BPA. And so what did we find? We found that chronic exposure to low-dose BPA increased the prostate stem cell numbers and increased the rate of proliferation of the progenitor cells. So in passage 3, when we looked at the total number of prostospheres that were derived per gland and also normalized to the input epithelial cells, when the data were decoded, we found that if they were exposed to phenylestradiol or the lowest dose of BPA, there was a significant increase in the total number of prostospheres. And this is representative of the stem cell numbers in that original tissue. The higher doses were too variable. We only had an N number of 3 and 5. But when we looked at the prostosphere sizes, we found a little bit of a different response, and the overall prostosphere size after 7 days of growth is representative of how fast those progenitor cells are proliferating in vitro. And here we found that if they were exposed chronically to ethinyl estradiol or to bisphenol A at the medium dose and somewhat at the higher 250 dose, there was a significant increase in progenitor proliferation. We then went on and did gene expression analysis for a basal progenitor pattern and a luminal progenitor pattern. And what we found with the decoded data was that if they were exposed to ethinyl estradiol or the 2 higher doses of BPA, there was a shift towards a basal progenitor pattern at the expense of losing a luminal progenitor pattern. So there was a lineage commitment shift. So to just summarize briefly, what we were able to find was that the lowest exposure of 2.5 micrograms of BPA significantly increased the number of prostate stem cells in the gland, whereas at 10 and somewhat at 100-fold higher doses, there was increased proliferation and a lineage commitment shift to basal progenitors. So we asked, how might the stem cell changes influence prostate cancer susceptibility? Well, we know that cancer risk is highly correlated to the number of normal stem cell divisions in most tissues, including the human prostate. We also know that tumor-initiating cells in prostate cancer are largely localized to the basal cell population. So we propose that this chronic in vivo low-dose exposure to BPA is increasing prostate stem cell numbers, altering lineage commitment, and this is underpinning an increased carcinogenic risk with aging. Thank you.
Thank you very much for that very interesting presentation, very sobering. Now we're going to go to Dr. Gail Prince. Sorry, Dr. Anna Soto is going to talk about the mammary gland and EDCs. And everyone, please remember to put your questions, I'm sorry, I made a mistake, on the Q&A at the bottom of your screen, and we'll address those later. Go ahead.
Hello, Dr.
Okay, I'm going, sorry,
Go.
I started talking in mute. This work has started 3 decades ago when Carlos Sonnenschein and myself discovered the first estrogen in plastic. Since then, we studied what types of effects would arise when fetuses and newborns are exposed to xenoestrogens, and our model is bisphenol A, obviously. And so during these 3 decades, we have found, before Clarity the following, that the mammary gland is exquisitely sensitive to exposure to BPA during fetal development. 25 nanograms per kilo per day is enough to produce deleterious effects. This exposure produces abnormal fetal, postpubertal, and adult development of the mammary gland. And finally, it induces mammary cancer. In addition, the effects of BPA during development sometimes mimic those of estradiol 17 beta, the ovarian estrogen, or the reference estrogen, ethinyl estradiol, but sometimes they are different. And estradiol and bisphenol A, both of them exhibit non-monotonic dose-response curves. So I'm going to show you some of this data that we gathered in those 3 decades. And one of those is exposure in utero to BPA. This is a fetal mammary gland. So we see the ductal system. And here is the control. At the same age, we look at the exposed to BPA, and we see that the mammary gland is much, the ductal system is much larger. On the right, we see the transcriptome of the tissue surrounding the epithelium here. We did that because it's there where the estrogen receptor is contained. And we see here that in VACO, we see here the blue is low expression, red is high expression. This pattern, BPA, changes it totally. Ethinyl estradiol is more similar to BPA than to VACO, but it's not exactly identical to BPA. So for each gene, it's a different response. Sometimes it's exactly the same. So if we look at 30 days of age, that is about 25 days after we finish exposure, the mammary gland is growing from here, a lymph node, invading the stroma. And so we see here a lot of invasion in the control. But when we look at the animal that had been exposed in utero to BPA, there is a delay on the invasion. That is one of the important phenomena of puberty. At 4 months of age, the mammary gland of the control at left has developed normally. That is, it's not very well differentiated. But then if we expose it to bisphenol A during fetal life, we see all these alveoli. And so this mammary gland looks like a mammary gland in the first third of pregnancy. And the animal would look like being pregnant since the 4th month of life to 9 months of age. Finally, if you look at this again, in some animals, cancer will develop. And here we see a whole mount that is a 3-dimensional mammary gland. And here we see a cut that is a histological section that shows that this tumor is identical to the tumors that humans show in the mammary gland, that is adenocarcinomas. So we interpret this phenomenon as follows. BPA will act in the brain and in the hypophysis, altering the pattern of hormone secretions, specifically the ones in the ovary. It alters cyclicity, and all these mammary, those hormones act on the mammary gland eventually. That is during puberty and later. BPA also acts in the mammary gland, the fetal mammary gland, altering its development and affecting the relation between the stroma and the epithelium. This results in this increased sensitivity of the mammary gland to the mammotropic hormones, and that would exacerbate the propensity to develop mammary cancer. Now, after this is in CLARITY, we propose to reproduce those endpoints, and we had to move from the CD mouse that has a simpler mammary gland to the rat mammary gland that is more complicated and more three-dimensional, requiring very special analysis that we had to develop. The first thing that we had to develop is an automated software for image reconstruction, because here is not a projection like a 2D, but a 3D reconstruction of the whole mammary tree. The software that we have to realize to walk through this mammary gland and measure every feature from branching angles to budding, etc., etc. Those are about 91 features. And then use of powerful statistical tools that would demonstrate or reject the hypothesis that these curves are non-monotonic. And this is the first time that permutation analysis was used in endocrine disruptor research. So I am going to talk now about those response curves. And that is important because in the regulatory experiments, that is the core CLARITY, the effects that are more important happen at low doses, and that is normal for all of us, the people that work on academia, but FDA has a problem with that, and that is that they expect that the curve will be monotonic. That is that as you increase the hormone, you will increase the response. And so this is showing the same sign all the time. Whereas when it's non-monotonic, it will grow, for example, for a while and then change sign, and that is, it will decrease. So the statistical treatment is different and more complex for the non-monotonic. So here is the clear demonstration that in the same animals that FDA used for the core study, we find a very clear non-monotonic dose response curve. It first goes up, then there is a break, and then it starts going up again. At the break between 25 and 250, we see that at 25, there is a lot of structures that we call buds, that is the origin of alveoli, more developed, and here is less developed, like the control. When we look at another parameter, that is the thickness of the epithelial tree, we see here that it's rather thick, whereas here is even thinner than in the control. So if we compare now the difference that there is between 25 and 250, those two doses, in most of the parameters that are 91, we find that this is the biggest gap, that is the most different phenotype between one and the other. And so if you look at this, this is all these parameters, and this is done at different ages, that is 21 days of age, 90 days, and 6 months. And you will see that in all the cases, yellow indicates the highest difference. In most of the cases, this happens between 25 and 250, showing that it's a recurrent pattern in the mammary gland, and therefore it's not due to random. When we do permutation analysis in order to ascertain that this is truly statistically significant, the original data shows this difference at all ages studied and in all the animals here. But if we do the permutations, that is to show what would happen in a random way, we see that those differences disappear. When we analyze those differences, and here is the real data at other intervals like vehicle and versus 2.5, etc., our data show that the difference is not significant between these doses. Therefore, it's clear that this is a very reproducible and statistically significant phenomenon. This pattern is not random, but represents a truly significant dose-related biological outcome. Regarding the difference or similarity between ethinyl estradiol and bisphenol A, we have observed both. Here we have bisphenol A producing the same effect than ethinyl estradiol, and both of them are different from control. Here we have the opposite. Here, control and ethinyl estradiol are the same, and bisphenol A produces an increase with respect to control and to ethinyl estradiol. So, our CLARITY studies reproduced our pre-CLARITY results in that bisphenol A induces abnormal development of the mammary gland, a phenomenon that we observe at all ages. Also, perinatal exposure increases the propensity to develop mammary cancer, as we found before. The effects of bisphenol A and ethinyl estradiol are not necessarily equal for all endpoints, sometimes are different. So not all estrogens are the same. And estradiol and bisphenol A induce non-monotonic dose response curves. Now, the most important effects here, as well as in the other PI studies, appear at the lowest doses. So, the conclusions. We have developed new tools and quantitative endpoints that could enhance the regulatory measurements, making them more accurate and more interesting too. When proper statistical methods are utilized, clear statistical evidence of non-monotonic dose response curves appear, just reinforcing the findings of all the PIs before these CLARITY studies. A breakpoint in our case between 25 and 250 nanograms per kilo indicates that this is not random because it happens practically at all the endpoints in the mammary gland. So it's just indicating that is a mechanism underneath the curve. That is a real biological phenomenon. And non-monotonic dose-response curves exist at all ages of the animal studies with the same breaking point. So now we compare this with what the FDA and the NCTR are concluding. So they say that the mammary cancer that they see at the lowest dose tested was due to random events. We now could say with confidence that that is not the case. The shape of the dose response curve indicates that the response is not monotonic, and therefore BPA induces cancer, and that is significant. Also, our study shows that different estrogens can produce either similar or very different effects. So therefore, their hypothesis, that is that because bisphenol A doesn't exactly replicate the effects of ethinyl estradiol, this is random, is not correct. And finally, these deleterious effects that we and others found appear mostly at the lowest dose tested. Hence, the tolerable daily intake, that is the so-called safe dose, has to be decreased accordingly. For the TDI in America, about 20,000 times, and for the one in Europe, about 2,000 times. Thank you for your attention.
Thank you very much, Dr. Soto, for this incredibly important research. Let's move now to Dr. Cheryl Rosenfeld, who's going to talk about studies across her endpoints and organs using her informatics research that she has done to make connections. And remind everyone to please put your questions into the Q&A box, and we will read those out, and so that everyone can hear the questions and their responses at the end of this session. So Dr. Rosenfeld, please begin.
Thank you, and I want to thank you for hosting us to talk about this important work that's come out. So as my colleagues have discussed, this consortium project included 14 independent investigators, and many of them received tissues from the same animal. And that's going to be an important point for this latest analysis I'm going to be talking about. And you've heard two of the published studies already from Dr. Soto and Dr. Prince, but many of these independent investigators, as Dr. Heindel discussed, have already published their independent results, and they clearly show that even low dose exposure to bisphenol A can lead to harmful effects in their particular system that they studied. But going back to one of the original goals of the study was to integrate the data, okay, and integrate the results across the studies. This has yet to be accomplished by our colleagues at the FDA and NTP, even though we have reminded them this is an important point. Around this time, though, my laboratory started using a novel informatics approach called mixomics analysis. And so herein, we discussed using this mixomics analysis, and we've used it in 3 ages: juvenile, young adult, and older adult, and 3 doses, the 2.5, the 250, and 250. And I want to point out this is microgram per kilogram, and I apologize for that mistake. So please note that it is indeed microgram per kilogram. Before I get into this mixomics analysis, though, I want to talk first about just correlation analysis in general, because this is important in terms of the studies we're going to be presenting. So some of you guys may have an idea of correlation analysis. Correlation, generally first off, we can consider 2 factors, okay? And again, you could consider, in this case, like the example is with auto. So different things about auto. So that, like if you look at auto mileage versus auto value, that would be considered a negative or inverse correlation. In all 3 of these cases, the negative, the 0, or the positive, it gives an R value. So in this case, the R value would be a negative R value. Now, if we look at the middle scenario, this could be auto body color versus auto quality. And again, the auto body color does not determine the auto quality, although some might differ. A red Corvette certainly is quite nice, but it doesn't affect the quality of the car itself. So that's would be our like 0 or 0.1 at least, but nothing significant. The last case scenario is an example where you see a positive correlation. This is where you get auto accidents. If you have more auto accidents, that would obviously increase your auto insurance. So you would have a positive value on the R. Unfortunately, life is not that simple in terms of just one factor against another. So generally we have where we are comparing many factors together. And in this example, this is one I wanted to present. It shows you then, in terms of, let's say, financial markets, okay? And I hope many of us may have already woken up and heard the Asian markets, European markets, how they're doing. Well, that's going to presumably affect then how our markets are going to do as well. So here showing these, positive and negative correlations. The positive are being shown in green, and the inverse or negative correlations in red. And what if financial markets across the globe can affect each other, so too can body systems. We have interconnections. We have circulatory systems. We have neural systems that connect each other, just like we have oceans and internet to connect the financial markets and determine how that goes. So I now want to present this mixomics analysis. And what it does, it basically integrates large data sets across independent studies. It was originally designed for like omics data. So again, we've got all these large omics data sets, but it can easily be applied for any data integration across independent studies. The other thing about mixomics analysis is great for doing heterogeneous and large data sets. And again, so we've used this independently in my lab, and I'll present some of those results to show you how we've used it. But we've been using this now for the past few years. And so when we were discussing this paper, it came to mind, well, wait a minute, we have the same animals across studies. Why not try to use this approach on a much broader scale? And this is, to our knowledge, the first use of this program to integrate data sets from multiple independent laboratories. And this is possible because that same animals, their organs were distributed to multiple investigators. So we could then tie it back in that sense. So I want to show you kind of what we have when we do these kind of mixomics analysis. There's two main types of data sets it gives you. The first is what we call a Diablo or R plot. Now, what you have to keep in mind is you may have general categories. And in our case, this is our example from our paper from last year. We were looking at the metabolome, the microbiome, and behavioral social and behavior vocalization, so how much they vocalize. And what you can see here is then, if we come on down, here's our metabolome to microbiome, and that would be the correlation of 0.86, so pretty highly significant. Again, if you look at some of the other ones, the microbiome to behavior social, 0.77, so again, pretty strong correlation. These are also the PCA plots, and you can see here in our example, we were doing a group with the AINs our control and genistein was our treatment, and you can see that they actually clearly separate in our PCA plots. So this is what it kind of shows in general. So this is for your general categories, but then you can get more specific because obviously, let's say within your metabolome, we looked at different metabolites, or in the microbiome, you have different bacterial changes. So again, we can then look at this at these individual factors, and this is called the Circos plot, and how individual factors within these categories, if it's a specific metabolite or a specific bacteria, and how they relate to those behaviors. And in this case, the red would indicate a positive correlation, blue would be a cold or a negative or inverse correlation. You can follow where they're going in terms of the different categories. The other thing this plot generally shows is that there's these lines at the edge right here. And whichever line is furthest away from the main circle, that means it has a greater value. So in this case, the control would have been greater for these particular categories than our treated group. So again, we're going to see how this then relates to when we can use it at a larger scale with the current CLARITY-BPA studies. Okay. So looking at this is our example of a female, and we had several of these that we included in our paper and also in supplemental material. And this is just one example. It gets very complicated fast, but again, keep in mind the same design holds. So this is your general R plot. These are your just general categories. In this case, we're looking at functions of white adipose tissue, ovarian follicles, heart results, mammary gland results, peptide hormones, steroid hormones, uterine results, and splenic function. And please keep in mind that these data were, again, each of these were generated in independent laboratories. These were all CLARITY-BPA investigators, but each of these were generated in their independent labs. But we were able to integrate them with this approach. And similar, again, you can see if you come in down, white adipose tissue, and white tissue weight, not surprisingly, that's a strong correlation. Here, if we look at mammary gland results and the peptide hormones, a pretty strong correlation at 0.72. So then we can delve in deeper with our circles plot and look at these. For instance, as Dr. Soto mentioned, she had 91 categories. So we can look at how her 91 categories within the mammary gland results relates to the other investigators' data. And that's the circles plot. And to make it a little bit easier in this case, again, this is with 6-month-old female data, same one as I was showing with the R plot. And at the lowest dose tested, 2.5 microgram per kilogram per day. But again, we have done now 3 doses and 3 different ages. And I wanted to then show you here in terms of we separated out the positive correlation in this circles plot and the inverse correlation in this one here. And this is going in deeper to each of these ones that people looked at. Like for instance, in splenic function, our colleague, Dr. Colbert, looked at several parameters related to immune function. And now you can see that some of these are crossing over and that they have a positive correlation to some of the other categories. In contrast, other ones show an inverse correlation. And so again, we can start getting ideas of how body systems relate to each other. Again, this is just the female data. Now, as I mentioned, we tested 3 different doses, 3 different ages. And I want to show you now the tables looking at 3 doses at the PND 180 at the 6 months old. And I want to show you something here. So what we've done in this case is this is the phenotype A to the phenotype B. And you can see this we are white adipose tissue weight, again, relative to some of the other categories, macrophages within white adipose tissue, relative to other categories, again, white adipose tissue weight with some of the other categories here. And what you will notice is this is our 2.5, the 250, and the 2500, and every one that's shaded, that means it's significant in that particular dose. And if you go across then, what you can see is, for instance, white adipose tissue weight is significant to splenic function at all 3 doses at this particular age. And this is just one age, but again, we've done the other 2 ages in a similar fashion. And some of the ones I found quite surprising. If you look at white adipose tissue weight related to splenic function, again, you see a strong correlation of all 3 doses at this particular age. So it's revealing these ones that might be significant and ones you had not thought about originally in terms of how they might relate to each other. Again, we've also seen just some other examples here. And one of these ones I want to point out here, again, this is ovarian follicles, so some of the other categories here, the heart results, mammary gland, and the peptide hormones. And one of the things I found really interesting was, in terms of these peptide hormones. And if you look, the peptide hormones show a strong correlation to splenic function, i.e. immune function, across all 3 doses. This is in the females, and as we'll see, we'll look at the males as well, but we're going to see a similar phenomenon when we look at the males. So again, immune functions can be regulated by all these circulatory factors, some of which we had not even considered to date. Here's our male data. For the male data, same age at the PND 180 at the lowest dose, 2.5. We have the white adipose tissue, the macrophages within the white adipose tissue weight, the heart results, peptide hormones, prostate results, and then splenic function. Again, these are just general categories, each of which then had several parameters that were examined within them. In the case of the males, we still see some strong correlations, but it's a little bit less stronger than the females, but we can still see some of these are coming up to about 0.7 correlation in terms of the R plot. We take a look now at the circles plot, where again, we delve in a little bit deeper. That's 2.5 microgram per kilogram, and it's the PND 180 for the males. And you can see, just like we saw with the females relative to the males, if you look here at the splenic function, we can see several connections to splenic function and ones you might not have thought about initially, and that includes even like prostate results. So it's something we had not even considered, maybe worth merit further pursuit in terms of how the immune function in the spleen relates to the prostate results. Again, strong positive correlations. And I want to point out, when we set these circles plots, anything considered about 0.7 is significant. We set this to 0.85, so highly stringent results. I mean, so if we have to be even less stringent, we may even see more correlations. We want to try to get, to balance the stringency with kind of just the background noise. So we've used a pretty stringent correlation here. And again, if we go to the inverse correlations, similar thing, we'll see that you can see many of these ones are inversely correlated. So again, as something goes up, something else might go down, or vice versa. If we look here, this is a table summarizing all 3 doses of BPA, the 2.5, the 250, and the 2500. I want to point out as well that we picked these 3 doses because these were the most common ones used across all the studies that we integrated here. Again, this is at the 180, but we've done similar ones in terms of the other 2 ages as well for the males. And again, you can see white adipose tissue, splenic function, very strong correlation here. Other thing, just like we saw with the females, the peptide hormones, very strong correlations across all 3 doses. One other thing that Dr. Soto mentioned as well for both the females and males, sometimes we saw ones that only showed at the low dose effect, at only the 2.5, and that's not surprising. Again, we know that this can be non-monotonic, and that's what can be revealed by then summarizing all these mixomics analysis together. So, this is how, when back to our physiology classes, when we think about bodily systems, this is your typical one. I remember this chart back to when I had Physiology 101. They show them as independent. You got your nervous system, the respiratory, circulatory, digestive, etc. And again, I also put in adipose tissue. Again, it's not really an organ system, but certainly can, as we've seen, can affect the other systems as well. So this is how we generally think of them. They act in, kind of like their own little world, but that's not the way they are. I mean, we know, again, they've got circulatory systems that connect each other, neural systems that connect each other. And so what I would like to hopefully people to take away from this type of analysis, and also that the fact that BPA can affect this is this type of system. And that's that these bodily systems are integrated together. And basically, if you impact one system, it's going to radiate out to other systems as well. And again, you're going to get systemic effects as a result. So again, just because we're looking at independent ones in each of our own studies doesn't mean that they don't have impacts on other ones. Our integrated results clearly suggest that that can indeed be the case. And to summarize, again, to our knowledge, the current work represents the first study using an informatics approach to integrate data across multiple independent investigator labs. Again, by using this whole body integrated approach, it reveals that low doses of BPA, the 2.5 dose, that are currently considered safe by the FDA can lead to simultaneous effects in multiple organ systems. And this is in both male and in female and at all ages that we examined. And I think to me, the most surprising correlations that came out from what we did was basically this integration between the immune system and almost all the other body systems. And this is quite scary in some ways because, again, it suggests if we impact the immune system, we're going to have detrimental consequences that will lead to, across the whole body. Again, I do want to point out, though, and this is something important, is that the main limitation of using this kind of mixomics approach, it only establishes correlation. But that's both, it can be, open up new avenues of research as well, because then we can try to figure out and tease apart which came first in terms of what are the precipitating factors that led to the systemic consequences. So it at least gives us then the next steps, the next avenue that we can test in that sense. With that, I'd like to kind of open it up back to our moderators.
Thank you very much, Dr. Rosenfeld. This is truly groundbreaking research and extremely important. Thank you very much for that. As Jerry Heindel mentioned earlier, there were some researchers who are not included in today's presentations for reasons of time, but we've asked Dr. Gail Prince to summarize some of that research that's not included in today's presentation and give us the take-home message. So Gail, do you want to go ahead and do your summary for us, please?
Sure. In the few minutes that are remaining, I just want to summarize some of the salient features that we come to in the conclusion section of this paper and the recommendations that we propose. So just again, to remind everybody that CLARITY-BPA was initially established to combine guideline and academic studies and together inform risk assessment on BPA safety. So in addition to the prostate gland and the mammary gland, which you heard today, there were a number of other different organ systems that were looked at, both in the guideline studies and in the academic labs. And in the academic labs, these were all specialists in their specific fields. Most of them had worked previously on BPA, on those end organs, and they used 21st century molecular and cellular approaches as opposed to the guideline studies, which are more focused on histopathology changes. So what did we find? Overall, we found a great deal of reproducibility amongst the studies when we compared them to previous studies by those laboratories and studies by dozens, if not hundreds of other investigators looking at the effects of BPA on their systems. There were some endpoints in some systems where the data was not reproduced, and there were different detailed endpoints where they were not identical. And we discuss this in the paper and provide a number of explanations for these. One is the experimental design was different than previous studies. For example, the use of gavage. The model system of the Sprague Dawley rat at the FDA facility and other model systems such as mice, which many previously used, and differing time points that were available for assessment in comparison to previous studies. But to really summarize it in a nutshell, in the findings in the CLARITY study, the conclusions remain the same. What we knew previously stands. Another big emphasis of our manuscript is sensitivity. In all of these organs listed here, we found reproducible endpoints in the CLARITY study as compared to what had been previously published. We find molecular and cellular endpoints that are far more sensitive than the standard guideline studies, and we recommend that these endpoints would be ideal for future EDC testing paradigms. I just want to emphasize again the low dose effect. In the majority of the studies, significant adverse effects were observed at the lowest dose, and this includes even in the guideline studies of 2.5 micrograms of BPA. The studies consistently observed non-monotonic, nonlinear responses. The responses were not the same between the different organs, but they were consistently non-monotonic, which is what we would expect from hormone-sensitive end organs. Importantly, the wonderful integrated mixomics analysis that was allowed because of this new study provided the ability to take the data analysis to a higher order of complexity. And in doing so, we find strong complex interrelationships across organs, including at the lowest dose. And so within the same animal, we're seeing system-wide responses to the lowest doses of BPA. So this provides very strong evidence that the low dose effects in individual organs are not spurious because they're systemic. There are many strengths to the study, which we discuss, and I want to point out that all the animals were treated at the FDA under guideline studies, GLP conditions. All the academic investigators were fully blinded to both the treatment group and the controls. And all the academic data was coded. And this coded data was locked down in a read-only file format before the codes were released to anyone for data analysis. And as such, we believe that all the academic studies meet the requirement for inclusion in risk assessment analysis. So prior risk assessment analysis depended upon guideline studies to reach the conclusions, and the ones done in the 1980s are still in use today. And there, these guideline studies found only high doses being toxic, and they derived the lowest observable adverse effective level at 50,000 micrograms per kg. And risk assessors utilize this to establish a NOAEL and a reference dose, also known as the acceptable tolerable intake, at 1,000-fold lower. And this has been going on for decades now, that 50 micrograms per kg per day of BPA is the safe reference dose. In the EU, it's now down to 4. Based on the CLARITY-BPA data, both in the guideline studies and particularly the academic studies, the new Lowell is unequivocally 2.5 micrograms per kg per day. And thus we argue that the reference dose needs and must have revisiting. If we set the new Lowell at 2.5 and continue to use a 1,000-fold lower factor in safety, the new acceptable intake dose would be 2.5 nanograms per kg, or 20,000-fold lower than what is currently utilized. So I encourage everybody to go back to the paper to get all the specifics, and we're now open for questions.
Thank you very much for that overview. That's very helpful. And we're at the end of our time, but we're going to take a few questions and just let everybody know that if we don't get to your question today, we'll make sure your questions are sent on to our presenters today, who will, time permitting, respond, and we will post the responses on the CHE website. So let's go quickly to just a couple of questions. The first one is from Pete Myers. He says, how does the 0.7 nanogram free BPA in rats compare to what's in people now? And this is a question for any panelist.
Well, there have been measurements. There's some controversy in terms of background contamination in BPA measurements. But there are very strong study measurements, particularly coming out of UCSF now, showing that humans in day-to-day exposure can be exposed to 1 nanogram per mil on a daily basis. Some in industrial settings, and infants can be exposed to even higher levels of BPA. So these levels that we're working with, 2.5 and 25 micrograms per kg, are definitely within the exposure values in day-to-day life. The higher doses certainly within industrial exposure levels.
I just wanted to echo Gail's comments as well. And so, you know, there was, in 2002, there was the only published study where they actually gave BPA to humans, the Vogel study. It's not been allowed since. And then at that point, the FDA said they couldn't detect anything. Well, it's a sensitivity issue. It clearly is a sensitivity issue. We have got more ways to now detect BPA. And I also want to bring everyone's attention that, you know, there's Pat Hunt's paper that just came out this year that suggests, again, we are underestimating the exposures and the internal concentrations. And I think that's a really important point. So again, I think to me, in our own studies, we have looked at, we've given similar kind of doses, and we have looked at the internal concentrations, and they replicate what we see in humans are unknowingly being exposed. And again, not deliberately like that one study, but just unknowingly be exposed. And I think as our sensitivity ways to measure BPA increases, I think we're going to find it's even probably more than we even think right now.
Thank you for that. Here's a question for Gail Prince from Dr. Laura Vandenberg, which was answered offline, but I think it's an important question, so I'll bring it up now. And Laura says, it looks like all the doses of BPA-induced PIN lesions. Would you argue that PIN lesions should be considered an adverse effect? Should regulators act on this, or is this a precursor to cancer, so more of a yellow than a red flag?
Prostate intraepithelial neoplasia, PIN lesions, if they're high-grade PIN lesions, are the immediate precursor to prostate cancer in humans. So they are an adverse effect. Whether low-grade move on that direction is unclear from the available data. But the important point in our studies was that the low-grade lesions with BPA exposure moved on to high-grade lesions. And so that's definitely an adverse effect. And this is in the context of adult exposure to estrogen.
All right. And how does the FDA justify not considering secondary exposures? The first thing a physician does when prescribing a new pharmaceutical is to ask, what are you taking already? And of course, drug interactions can be highly deleterious.
Well, under standard guideline studies, they don't do secondary exposures. So they only look at the primary effect of the agent. And so that's all they would consider when they came up with their analysis. They strongly argued against my ability to put T&E pellets into these animals, and we prevailed, but they didn't want to do it.
the two-hit hypothesis, though, is gaining a lot of traction. I mean, both like with what Gail's done with her study and also even like other extrinsic factors. So they've looked at the combination of exposures to EDCs and later stress, and they can show that they show exacerbated responses. I mean, this idea of the two hits, not, you know, it clearly is a phenomenon, and I think it merits further work that, you know, basically what we're exposed to in utero life or during development or early on can lead, you know, can make us susceptible to later consequences. And I think, to me, we need to understand that phenomenon better.
Yeah, the two-hit hypothesis is the foundation of cancer, so that's well established.
Okay. Many products are now made with BPS instead of BPA. Does this research have implications for exposures to BPS as well?
Yeah.
Certainly.
We just published a study where we looked at both BPA and BPS and effects in the placenta and the mouse, and they replicate each other. I mean, the findings are almost identical. And others have shown that BPS can bind into the same receptors. In fact, for some of them, they show higher affinity. So I think, to me, a lot of the studies that we are, you know, things we're finding about BPA hold for BPS and the other BPA analogs as well.
And Sue Fenton has shown that also induces, I mean, the replacements S and bisphenol AF also produce deleterious effects in the mammary gland, including tumors.
Right, here's from Eva Kumar. My question is related to Girona et al.'s article, BPA. The title of the article is BPA Have Flawed Analytical Techniques Compromise Risk Assessments, which was published in the Lancet Diabetes and Endocrinology Journal. In your view, has the risk to human health from BPA been dramatically underestimated due to analytical challenges?
Yes, I agree.
Okay.
No doubt about it.
That's the paper I was referencing.
Yeah, that's from UCSF.
Mm-hmm.
Okay. Okay. And then I'm intrigued with the application of findings the public need. Someone says she's no expert, and many aren't, of course. Is there anything in any way to remove or neutralize BPA in products or in ingested BPA?
Remove is the only way. So you have to regulate in order not to use it in the applications it's used today in order to decrease exposure.
I mean, BPA is rapidly cleared from the body. It doesn't hang around for a long period of time at all. And so the answer is to stop using products that contain it.
Okay. And then someone has a question about the mechanism driving the brake detoxification. This is from Krista McCoy. So the body sees the high dose as a toxin, detoxifies it, but not completely because there's still effects. And then as doses increase, detox is less helpful. Anyway,
I think that
what drives the brake? Very interesting.
I think that what happens there, if you look at what is underneath, is that when you have a complex non-monotonic dose response, is the sum of two or more monotonic dose responses. And that we have shown with Carlson and Shine, that if you have a cell line that has an inverted U-shaped dose response curve, then you can obtain variants that have the first part or the second part. And not only that, the genes involved in the first part are different from the ones involved in the second part. And I think that regarding bisphenol A, that is clearly shown in papers by Angel Nadal's group, where he shows that some of the effects go through alpha and some through beta, estrogen receptor alpha and beta in the pancreas, and that could be totally separated. So it's a sum of the two types of effects. That is from the viewpoint of what is causing that. You can separate down. I don't think that we have to think of toxicity, but simply that biology is complex and that we are at different doses, we are triggering different processes.
I'd like to mention as well that this whole idea of detoxification, we have to keep in mind as to when the exposure occurs, because during fetal life, the fetus liver, where it would have to get detoxified, doesn't really have the enzymes. So again, the fetus is really susceptible to early exposures because it cannot metabolize it to the same extent an adult can. And these studies, most of, again, they were giving during this, perinatal life when the enzymes are just not there. So again, I think they're even more vulnerable than if the exposure were later in life.
Okay, thank you very much. I think we've gone to our limit for today's questions and answer period. So once again, I want to thank the researchers for their excellent presentations and also for their incredibly important research in this topic. I hope that the FDA and other government agencies begin to pay attention. It's critically important. I'm going to turn the webinar now over to Hannah to do our formal exit.
Great. Thank you so much, Cheryl, and to all of our speakers today. We're approaching the end of today's webinar. A video recording will be available on SHEA's website soon, and tomorrow you'll receive an email containing a link to the video. The next SHEA Partnership webinar will take place tomorrow and is titled Disparities in Cancer. This is our final webinar in our Cancer and Environment series. To learn more and RSVP, please visit our website at healthandenvironment.org. If you are new to SHEA and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these SHEA Partnership webinars bringing you the latest environmental health research for free, we encourage you to support SHEA's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would also like to remind you that if you had any questions we didn't have a chance to answer during the Q&A portion of the webinar today, please submit it to Cheryl Patton at the email listed in our final slide on the page by July 28th, and we will respond via email. With that, I would like to thank our speakers, Dr. Heindel, Dr. Prinz, Dr. Soto, and Dr. Rosenfeld for taking time to present today and for all of your amazing research and work. And to Cheryl for your excellent moderation. Thank you so much for joining us. We're wishing all of you health and wellness. Have a great day.